We just found aliens, not in some distant galaxy billions of light years away, not on Mars where rovers have been searching for decades, not even in the clouds of Venus where some scientists speculate microbial life might exist. We found them right here on Earth, hiding in a place so hostile, so extreme, so utterly inhospitable that it might as well be another planet entirely. 6,000 meters below the surface of the ocean, deeper than Mount Denali is tall, in a trench where tectonic plates are literally colliding with the force of continents, scientists have discovered creatures so bizarre, so fundamentally different from anything in our experience, that they challenge everything we thought we knew about the limits of life. Picture this: a clam that does not filter feed like every other clam on Earth, but instead hunts like a predator, using a specialized hood to vacuum up prey in the pitch-black abyss. An isopod covered in bumps that look exactly like popped kernels of popcorn, living as a parasite inside the gill chamber of a ghost shrimp, a mollusk from a class of animals that was supposed to have gone completely extinct 375 million years ago, before the first dinosaurs, before the first reptiles, back when life on land was still figuring out how to breathe air. These are not science fiction creations dreamed up by a screenwriter. These are not computer-generated aliens from a Hollywood blockbuster. These are real animals, living right now at this very moment, in conditions that would crush a military submarine like a tin can and kill a human being in seconds. And in November of 2025, a team of more than 20 international researchers working under the Senckenberg Ocean Species Alliance unveiled 14 of these alien species to the world, along with compelling proof that we have been studying deep-sea life all wrong for the past century. They showed us that we do not need to destroy these creatures to understand them. They proved that modern technology can give us unprecedented insights into anatomy and biology without ever making a single cut. This is the story of what we found when we finally learned to look without destroying what we were looking for. This is the story of the methodology revolution that is changing marine taxonomy forever. This is the story of the aliens at 6,000 meters deep and why they matter more than you might think. Let us talk about the Aleutian Trench, because this is where our story really takes place. You have probably never heard of it, and honestly, that is exactly the problem. While everyone obsesses over the Mariana Trench because it has the deepest single point on Earth, the Challenger Deep at nearly 11,000 meters, the Aleutian Trench has been quietly hosting some of the most extreme and bizarre life on the planet, operating in relative obscurity. It stretches for an astounding 3,400 kilometers along the southern coast of Alaska and out into the Aleutian Islands, forming a massive geological scar in the ocean floor where something violent and fundamental is happening every single moment. The Pacific Plate and the North American Plate are colliding at a rate of about 7.5 centimeters per year. Now, 7.5 centimeters might not sound particularly dramatic. That is about 3 inches. You could measure that with a ruler, but when you are talking about continental-scale slabs of rock, each thousands of kilometers wide and dozens of kilometers thick, moving with the accumulated force of entire continents behind them, 7.5 centimeters per year becomes catastrophic. Imagine 2 freight trains, each the size of Asia, moving toward each other in slow motion. That is what is happening in the Aleutian Trench. The Pacific Plate is being shoved underneath the North American Plate at an angle of nearly 45 degrees in a geological process called subduction. This is not a gentle meeting of neighbours. This is not a polite boundary. This is a geological demolition derby happening in the slowest, most powerful slow motion you can imagine. The descending Pacific Plate drags sediment, water, organic material, and entire chunks of ocean floor down into the Earth's mantle, where temperatures climb into the hundreds and thousands of degrees Celsius, and pressures become incomprehensible to the human mind. The friction between these plates is what causes the massive earthquakes that periodically devastate Alaska and send tsunamis racing across the Pacific. The 1964 Alaska earthquake, one of the most powerful earthquakes ever recorded at magnitude 9.2, was caused by this subduction zone. The resulting tsunami killed over 130 people and caused damage as far away as Hawaii and California. But we're not here to talk about earthquakes. We're here to talk about life. The deepest part of the Aleutian Trench plunges to 7,822 meters below sea level, located about 145 kilometers south-southwest of Buldir Island, a tiny remote island in the western Aleutians that most people could not find on a map if their lives depended on it. At that depth, The pressure is 732 atmospheres. To express that in units that might mean something, that is over 10,700 pounds per square inch. To put that in perspective, if you were somehow able to survive long enough to stand at the bottom of this trench, the weight of the entire ocean above you would be pressing down with a force equivalent to having 50 fully loaded Boeing 747 jumbo jets stacked on top of every single square inch of your body. Your lungs would collapse instantly, your bones would shatter, your cells would be crushed. This is not an environment designed for human life, and it is cold. The water temperature hovers just above freezing, typically between 1 and 4 degrees Celsius. There are no warm currents this deep. There is no sunlight to provide warmth. The ocean down here is as close to frozen as salt water can get without actually forming ice, and it is dark. Not just dark like nighttime, not dark like a cave, dark like the void of space, pitch black, absolute, total, complete darkness. No sunlight has penetrated to these depths for billions of years, not since the sun first started shining. If you turned on a flashlight at the bottom of the Aleutian Trench, you would be the first light source that water has seen since the Precambrian. By every logical measure, this should be a sterile, lifeless void. This should be an aquatic desert where nothing can survive. The combination of crushing pressure, near freezing temperatures, and complete darkness should make complex life impossible. And yet, life has found a way, not just to survive, but to thrive in what we would consider hell. Not just to scrape by, but to diversify, to specialize, to evolve into forms that are more bizarre than anything we see on the surface. And that is where our story of the November 2025 discoveries really begins, because what lives down there is rewriting our understanding of what life can be. The absolute star of our alien lineup, the creature that deserves top billing in this deep-sea horror show, is something called Meionera aleutiana. It is a bivalve, which means it belongs to the same broad group as clams, oysters, mussels, and scallops. But calling this thing a clam is like calling a great white shark a fish, or calling a wolf a dog. Technically true from a taxonomic standpoint, but it misses the entire point and fails to capture just how different this creature really is from its relatives. Most clams, the vast majority of the roughly 15,000 bivalve species on Earth, are peaceful filter feeders. They have a simple lifestyle perfected over millions of years of evolution. They sit in the sand or mud, partially buried, and they open their shells just slightly, maybe a centimetre or two. Then they pump water through their gills using tiny hair-like structures called cilia. As the water flows through, the gills capture microscopic particles of food, phytoplankton, bacteria, bits of organic detritus drifting down from the surface. The gills are incredibly efficient filters, capable of catching particles as small as a few micrometers. The clam processes thousands of litres of water every day, extracting the tiny nutritious bits and expelling the clean water back into the environment. They are the vacuum cleaners of the ocean floor, the water treatment plants, the passive collectors of whatever drifts by on the current. It is an elegant survival strategy that requires almost no energy expenditure and works brilliantly in environments where food is abundant and evenly distributed. But Meionera aleutiana does not do any of that. Meionera aleutiana is a carnivore. It hunts. Let that sink in for a moment, because this is genuinely shocking to anyone who knows bivalves. A clam that eats meat. A mollusk that actively pursues and captures prey. This is not supposed to exist in the natural world, and yet here it is, living at depths between 5,170 and 5,280 meters in the eastern part of the Aleutian Trench, and it has evolved a completely different feeding strategy from every other bivalve most people have ever encountered. Instead of filter feeding, Meionera aleutiana has a specialized muscular structure called a hood that extends from the modified edges of its mantle, the fleshy tissue that lines the inside of its shell. Think of this hood as something like a vacuum cleaner attachment, a flexible tube or funnel that the clam can extend outside its shell. When a tiny crustacean, perhaps a copepod or amphipod, or some other small invertebrate gets close enough, detected by chemical sensors or mechanical vibrations that we do not yet fully understand, the hood activates. It creates a sudden suction current, a localized water movement that pulls the unsuspecting prey directly into the clam's mouth. This is active predation. This is not waiting for food to drift by. This is hunting. This is a clam behaving like a miniature predator in an environment where every calorie counts and where sitting around waiting for food to float past might mean starving to death. The evolutionary pressure that drove this adaptation must have been intense, because filter feeding is a much simpler strategy. Evolution does not usually take the complex route when the simple one works. But in the deep ocean, especially at depths of 5,000 meters and below, food is incredibly scarce. The rain of organic material from the surface, what marine biologists call marine snow, becomes thinner and thinner the deeper you go. Most of it gets eaten by animals living in shallower zones. By the time anything reaches 5,000 meters, it is mostly skeletal fragments and the occasional large carcass that sinks quickly, like a dead whale. For a filter feeder, this means waiting potentially days or weeks between meals. But for a carnivore that can actively capture live prey, the game changes. Small crustaceans and worms live in the sediment, feeding on bacteria and whatever organic material they can scavenge. They are concentrated sources of nutrition, little packets of protein and fat, and if you can catch them, you can survive where a filter feeder might starve. That is what Meionera aleutiana has figured out. It has carved out an ecological niche that other bivalves cannot exploit, and in doing so, it has become something genuinely alien. The shell itself is a work of bizarre art that would not look out of place in a science fiction film. It is thin and fragile, almost translucent when you hold it up to a light source, with an olive green colour that seems completely out of place for a deep-sea animal. Most organisms living in the permanent darkness of the abyss are pale, white, or colourless. There is no light down there, so there is no evolutionary advantage to pigmentation. Colour costs energy to produce, and in an environment where every calorie is precious, why waste resources on something that serves no purpose? But Meionera aleutiana has colour. That olive green tint suggests the presence of pigment molecules, and while we do not yet know why this clam has retained its colour, it is one more mystery about this species. The shell surface is covered in what the researchers describe as feather-like plates called lamellae, delicate structures that overlap like shingles on a roof, along with small bumpy protrusions called pustules scattered across the surface. If you were served this clam at a seafood restaurant, you would almost certainly send it back, possibly lodging a complaint with the health department. It looks fragile, delicate, gossamer, almost like a piece of art made from rice paper rather than a living animal that survives under crushing pressure. And that fragility is deceptive, because this shell has to withstand over 700 atmospheres of pressure, roughly 10,700 pounds per square inch. How does it do that? The answer is counterintuitive but elegant. At these depths, the pressure inside the organism equals the pressure outside. Water is incompressible, and cells are mostly water, so there is no pressure gradient trying to crush the clam. The organism does not experience pressure the way we would if we were suddenly transported to 5,000 meters depth. For the clam, this is normal. The pressure is everywhere, inside and out, perfectly balanced. What matters is not resisting the pressure, but maintaining cellular function despite it. Proteins have to fold correctly, enzymes have to catalyze reactions, cell membranes have to remain fluid. These are the real challenges, and Meionera aleutiana has solved them through biochemical adaptations we are only beginning to understand. That thin shell is not a weakness. It is optimized for an environment where heavy armor is unnecessary. Why build a tank when you do not need one? This discovery also set a significant depth record for the entire Meionera genus, and depth records in marine biology are a big deal because they represent the absolute limits of where life can exist. Previously, the deepest known member of the genus Meionera was found at 4,400 meters in the Tasman Sea in the southwest Pacific. That was already impressively deep. But Meionera aleutiana was collected from depths of 5,170 to 5,280 meters, roughly 800 meters deeper than any previously documented individual from this genus. 800 meters might not sound like much when you're already talking about thousands of meters, but in the deep ocean, every 100 meters is a new world with new challenges, new, selective pressures, new opportunities, temperature drops. Pressure increases by about 10 atmospheres for every 100 meters of depth. Food availability changes. The composition of the sediment shifts. To find a species thriving 800 meters deeper than anyone thought possible for its genus tells us that these trenches are not uniform environments, but incredibly complex, layered ecosystems with multiple niches, microhabitats, and ecological opportunities that we are only just beginning to understand. And it raises an obvious question. If Mayanera can live at 5,280 meters, what is stopping it from going even deeper? What are the actual limits? We do not know yet. And that is exactly why discoveries like this matter. Now here is where things get revolutionary, where the science itself becomes as fascinating as the creatures being studied. In the old days of marine taxonomy, which is to say pretty much every decade before this one, if you wanted to study a new deep-sea species and describe it for science, you had to cut it open. You would take your rare, incredibly precious specimen that you had just dragged up from the abyss at enormous expense and effort, requiring a research vessel that cost tens of thousands of dollars a day to operate, sophisticated sampling equipment, expert technicians, and often months of planning and travel, and you would dissect it. You would destroy it to understand it. You would take scalpels and scissors and carefully slice through tissues, documenting every organ, every muscle, every structure as you went, making detailed drawings or photographs, taking measurements, preserving bits of tissue for later analysis. The process could take days or even weeks for a single specimen, and at the end of it, the animal was gone, reduced to tissue samples floating in jars of ethanol, and a set of anatomical drawings that could never fully capture the three-dimensional complexity of the living organism. That was just how taxonomy worked for over 2 centuries. It was destructive by necessity because there was no other way to see inside an organism without cutting it open. But with Mayanera aleutiana, the researchers did something fundamentally different, something that represents a paradigm shift in how we study biodiversity. They used a technique called micro-CT scanning, and it changed absolutely everything about what is possible in modern taxonomy. Micro-CT stands for microcomputed tomography, and while the name is a mouthful, the concept is essentially the same technology as the CAT scans used in hospitals when a doctor needs to see inside your body without cutting you open, just scaled down and refined for tiny biological specimens that might be only a few millimeters long. Here is how it works, and this is genuinely fascinating stuff. The specimen is carefully positioned on a rotating platform inside a machine that looks somewhat like a miniature version of a medical CT scanner. The machine has an X-ray source on one side and a highly sensitive detector on the opposite side. The X-ray source fires a beam of high-energy photons through the specimen. As these X-rays pass through the biological tissues, they are absorbed to different degrees depending on what they encounter. Dense materials like calcium in shells and skeletal structures absorb more X-rays. Soft tissues like muscle and nervous tissue absorb an intermediate amount. Watery structures and empty spaces absorb very little. The detector on the far side measures exactly how many X-rays made it through and how much they were attenuated, which is a fancy way of saying reduced in intensity. But one cross-section is not enough. One slice through the organism would give you limited information, like looking at a single frame from a movie and trying to understand the entire plot. So the specimen rotates. The platform turns by a tiny fraction of a degree, maybe half a degree or even less, and another X-ray image is captured. Then it rotates again, and again, and again. Hundreds or thousands of rotations, each one providing a slightly different view, a new angle through the three-dimensional structure of the organism. By the time the specimen has completed a full 360-degree rotation, the computer has collected hundreds of two-dimensional X-ray images, each one representing a different perspective through the body. Now comes the computational magic. Sophisticated algorithms using mathematical techniques developed originally for astronomy and particle physics reconstruct these hundreds of two-dimensional images into a complete three-dimensional model. They do this through a process called tomographic reconstruction, which is essentially working backwards from all those different X-ray shadows to figure out exactly where every piece of tissue must be in three-dimensional space to produce the pattern of shadows observed. The result is astonishing. You end up with a digital model of the organism that you can manipulate on a computer screen. You can rotate it. You can zoom in. You can digitally slice through it at any angle, not just the standard anatomical planes, but any arbitrary plane you choose. You can peel away layers virtually, removing the shell to see the soft tissues inside, removing the muscles to see the digestive system, removing everything except the nervous system to see how the nerves are distributed through the body. You can make different tissues invisible or transparent. You can color code structures. You can take measurements with sub-millimeter precision. You can count features, analyze shapes, compare structures across different specimens, all of this without ever making a single physical cut. For Meionera aleutiana, this process generated over 2,000 individual tomographic images. That is 2,000 virtual slices through the body of this clam, each one revealing another layer of its internal anatomy in stunning detail. The researchers could see the digestive tract from the mouth all the way through to the anus. They could see the gill structures, those modified gills that are probably involved in respiration but not in filter feeding. They could see the muscular system, including the specialized muscles that operate the predatory hood. They could see the nervous tissue, the reproductive organs, the crystalline style that helps with digestion, the hepatopancreas that functions somewhat like a liver, and pancreas combined, all the tiny anatomical details that define this species and make it different from every other known bivalve. And the specimen itself remained completely intact throughout the entire process. This is only the second time in the history of marine biology that a bivalve species has been fully described using solely non-invasive micro-CT scanning, with no dissection whatsoever. The first time proved it could be done and established the methodology. The second time, with Meionera aleutiana, proved it should be done, that this should become the new standard for how we study rare and precious specimens. Think carefully about what this means for the future of marine taxonomy and biodiversity research. Every deep-sea expedition is expensive, risky, weather-dependent, and limited in scope. Every specimen collected represents a tiny sample of a vast ecosystem we can barely access. In some cases, we might only ever collect a single individual of a species. That specimen is irreplaceable. It might be decades before another expedition goes to that exact location at that exact depth. It might never happen. In the traditional approach to taxonomy, describing a new species meant destroying the holotype, the single official specimen that serves as the reference point for that species for all of science for all time. Once you cut it open, once you separate all the tissues and organs and preserve them in different solutions, you can never put it back together. Future scientists can look at the preserved tissues, sure, but they cannot see the organism as a complete integrated whole. They cannot re-examine it using techniques that have not been invented yet. They are limited by the decisions made by the original describer, by what that person chose to measure, chose to photograph, chose to preserve. But with micro-CT scanning, everything changes. The holotype remains preserved, intact, whole, available for future researchers to study with even better technology that we have not dreamed up yet. Maybe in 20 years, someone will develop an imaging technique that can reveal chemical compositions of tissues. Maybe in 50 years, someone will figure out how to extract genetic information from specific cells without destroying them. Maybe in 100 years, technology we cannot even imagine will let us reconstruct the molecular structure of proteins directly from preserved specimens. If the holotype still exists as a complete organism, all of that remains possible. We are no longer destroying aliens to understand them. We are learning to observe them, to document them, to appreciate them without ending their existence, even if that existence is now preserved in ethanol rather than lived in the ocean. This is the future of taxonomy, and Meionera aleutiana is proof that it works, that it is practical, that it produces results that are every bit as detailed and scientifically rigorous as traditional dissection methods. And that matters enormously for conservation, for education, for our relationship with the natural world. If the carnivorous clam Meionera aleutiana is the star of this show, the headliner that gets top billing and draws the crowds, then Zeaion averta is absolutely the breakout character that steals every scene it appears in, the quirky sidekick that audiences remember long after the credits roll. This is an isopod, which means it belongs to the crustaceans, a massive and diverse group that includes everything from crabs and lobsters to pillbugs and the little roly-poly bugs you find under rocks in your garden. But Zeaion averta looks nothing like the harmless little pillbugs that curl into balls when you touch them. The female of this species, and this detail is important because males look completely different, has raised protrusions covering the entire dorsal surface of its back, bumps and knobs and irregular bulges that look exactly, and I mean exactly, like popped kernels of popcorn. Not kind of like popcorn in a vague, if you squint it might resemble way, not vaguely reminiscent of popcorn if you're feeling generous with your imagination. These bumps look exactly like popcorn, like someone took actual popped kernels, shrunk them down to isopod scale, and glued them onto this creature's back in a random pattern. The resemblance is so striking, so unmistakable, so perfect, that the scientist who discovered and described this species decided to name the entire genus after corn. The name Zeaione comes from Zea, which is the scientific genus name for corn, for maize, for that yellow grain that Native Americans domesticated thousands of years ago and that now grows in vast monoculture fields across the Midwest. This thing is literally the corn isopod, the popcorn crustacean, a name so descriptive and so whimsical that it sounds like something from a children's book rather than a peer-reviewed taxonomic journal. But the popcorn appearance, as visually striking and memorable as it is not even close to the weirdest thing about Zeaione averta. The appearance is just packaging. The real horror show, the genuinely disturbing part, is what this creature does for a living. Zeaione averta is a parasite, and not just any parasite, but a body-snatching, space-invading, home-stealing parasite that would be right at home in the Alien franchise. It lives inside the gill chamber of a ghost shrimp, a fairly odd animal in its own right. Ghost shrimp, in case you have never encountered one, are semi-transparent burrowing crustaceans that live in muddy or sandy sediments in coastal areas. They are called ghost shrimp because you can literally see through them. Their bodies are translucent, sometimes almost completely clear except for a few visible internal organs, and that give them an ethereal spectral quality. They spend most of their lives underground, digging elaborate tunnel systems through the sediment, sometimes creating burrows that extend down 30 or 40 centimeters with multiple chambers and passageways. They are ecosystem engineers, animals whose burrowing activity fundamentally alters the environment around them, increasing oxygen penetration into the sediment, mixing nutrients, creating habitat for other species, and somewhere in those dark, twisting tunnels, Zeaione averta sets up shop. The life cycle, as far as we understand it, goes something like this. A larval or juvenile isopod, tiny and mobile, finds a ghost shrimp burrow. How it finds the burrow is not entirely clear. Maybe it follows chemical cues in the water, maybe it detects vibrations from the shrimp's digging. Maybe it just wanders randomly until it gets lucky. But once it finds a burrow, it crawls inside. It makes its way through the tunnel system until it encounters the shrimp. And then, in a move that would make any horror movie villain proud, it crawls into the shrimp's gill chamber. The gill chamber is the space inside the shrimp's carapace where the gills are located, the organs the shrimp uses to extract oxygen from water. It is a vital space, arguably the most important part of the shrimp's respiratory system. And Zeaione averta just moves in, squeezing itself into this tight space, wedging itself between delicate gill filaments, and staying there. Permanently. It is not a temporary visit. It is not a brief intrusion. This is a full-time occupancy, a permanent invasion, a hostile takeover of prime real estate inside another animal's body. The isopod lives there, feeding on something, either particulate food that the shrimp brings into its burrow or possibly, and this is less certain, feeding directly on the shrimp's tissues or bodily fluids. The parasitology is not completely understood yet because observing what happens inside a ghost shrimp burrow 30 centimeters underground is technically challenging, but the relationship is clearly exploitative. The isopod benefits. The shrimp suffers, though probably not fatally, at least not immediately. That would be terrible strategy for a parasite. If you kill your host quickly, you lose your home and your food source, and you die along with it. Good parasites, from an evolutionary perspective, keep their hosts alive as long as possible, extracting resources slowly, taking just enough to survive without causing catastrophic damage. Zeaione averta seems to follow this rule. The infected shrimp continues to live, continues to burrow, continues to function, but now it has a passenger, a freeloader, a body, snatcher living inside its respiratory system and looking for all the world like someone spilled popcorn inside a crustacean. Now here is the interesting part, the detail that might seem contradictory given the title of this video about aliens at 6,000 meters deep. Zeaione averta was not found at 6,000 meters. It was not even found in the deep sea. It was discovered in the Australian intertidal zone, which means the area of coastline between the high tide mark and the low tide mark, the strip of habitat that is underwater during high tide and exposed to air during low tide. This is practically the surface compared to the crushing depths of the Aleutian Trench. It is about as far from 6,000 meters as you can get while still being in the ocean. So why are we talking about it in a video focused on deep-sea aliens? Why include it in this lineup alongside creatures from the abyss? Because it was part of the same November 2025 batch of discoveries published by the Senckenberg Ocean Species Alliance in their Ocean Species Discoveries collection. This compilation of 14 new species was never meant to be exclusively about the deep sea. It was designed to showcase the full range of marine biodiversity discovery, from the shallowest tide pools to the deepest trenches, demonstrating that the ocean at every single depth, at every pressure, at every temperature, at every level of light penetration, is full of bizarre and undiscovered life forms that challenge our assumptions about what is normal. Zeonevita might live in shallow water, in an environment that you could theoretically visit with just a snorkel and a willingness to dig in the mud, but it is no less alien than anything from 6,000 meters down. If anything, the fact that we are still discovering completely new genera of parasitic crustaceans in the intertidal zone, in habitats that are literally within wading distance of populated beaches, tells you just how little we know about marine biodiversity, even in the most accessible parts of the ocean. And honestly, if you told someone, anyone, that scientists had discovered a popcorn-covered parasite that lives inside a ghost shrimp, and that all of this is real and happening right now on Earth, not in some distant galaxy or in a science fiction novel, but in actual tide pools you could visit on your next beach vacation, they would probably assume you were describing a rejected Pokemon or a creature from a particularly creative science fiction novel. But it is real, it is alive, it is reproducing, and it has been here the entire time, for millions of years probably, following this bizarre parasitic lifestyle generation after generation, completely unknown to science until 2025, when someone finally looked closely enough at the right ghost shrimp in the right mudflat in Australia and said, "What on earth is that popcorn-looking thing?" Now we come to perhaps the most philosophically profound discovery of the entire collection, the one that makes you rethink what we mean when we say a species is extinct. Valeropolina gretcheni. This specimen was pulled up from 6,465 meters deep in the Aleutian Trench, south of Unalaska Island, making it one of the deepest living animals described in this entire study, and putting it well into what marine biologists call the Hadal Zone, the deepest parts of the ocean named after Hades, the Greek underworld. And it is not just any mollusk sitting at the edge of the abyss. It is a Monoplacophoran, a member of a class of animals that science had declared completely extinct 375 million years ago. Let me emphasize that timeline again because it is almost incomprehensible. 375 million years ago, the Devonian period. For context, that is not just before the dinosaurs. That is before the first reptiles evolved. That is before the first insects developed wings. That is back when the most advanced life on land consisted of amphibians that had only recently crawled out of the ocean and were still figuring out the whole breathing air situation. The first forests were just beginning to appear, primitive plants barely taller than a person spreading across landscapes that had been barren rock for billions of years. The continents were arranged in configurations we would not recognize, with Gondwana and Laurussia as the major landmasses. And in the oceans, Monoplacophorans were swimming around, or rather crawling around, because these are not swimmers but slow-moving bottom dwellers, living their lives completely unaware that in the distant future, paleontologists would think they were extinct. Monoplacophorans first appear in the fossil record way back in the Cambrian period, over 500 million years ago during that extraordinary explosion of biodiversity when most modern animal phyla suddenly appeared in the geological record over a relatively short span of time. They were among the first mollusks, simple creatures with cap-shaped shells covering simple bodies, crawling across the ancient seabed eating whatever organic material they could scrape up with their radulae. The ribbon-like structures covered in tiny teeth that mollusks use for feeding. For hundreds of millions of years they thrived, diversified into multiple species, left behind countless fossils in marine sedimentary rocks all over the world. And then, around 375 million years ago, they vanished from the fossil record. Paleontologists searched for Monoplacophoran fossils in rocks younger than the Devonian and found nothing. The logical conclusion was that the group had gone extinct, probably wiped out in one of the periodic mass extinction events that have punctuated Earth's history, joining countless other lineages that flourished for a time and then disappeared forever. For the entire 20th century, that was the accepted, scientific consensus. Monoplacophorans were extinct, dead, gone, lost to deep time. Interesting as fossils, certainly, important for understanding early mollusk evolution, but no longer part of the living world. Except they were never extinct. They were just hiding in the deep ocean where nobody was looking. On May 6, 1952, one of the most dramatic discoveries in the history of malacology, which is the scientific study of mollusks, occurred off the coast of Costa Rica. A Danish deep-sea research expedition called the Galathea Expedition was trawling the ocean floor at a depth of 3,590 meters, dragging nets across the sediment in the kind of exploratory sampling that was cutting-edge oceanography in the 1950s. When they hauled up the net and sorted through the mud and sediment and debris, they found 10 small cap-shaped shells, each only about 3 centimeters long. The shells looked odd, different from anything the researchers were familiar with. They did not quite fit into any known modern group, and when they examined them more closely, when they looked at the muscle scars on the inside of the shells and the arrangement of the soft body parts in the few specimens that were intact enough to examine, they realized with mounting astonishment that they were looking at living monoplacophorans, creatures that were supposed to have been extinct for 375 million years. The discovery was a shockwave through the scientific community, one of those moments when everyone has to revise their understanding of what is possible. It was like finding a living trilobite or a living dinosaur. The lead researcher, a Danish zoologist named Henning Lemche, formally described the species 5 years later in 1957, naming it Neopelina galatheae after the expedition ship, and these specimens were immediately hailed as living fossils, a term popularized by Darwin to describe organisms that appear virtually unchanged from their ancient ancestors, evolutionary relics that somehow survived when all their relatives went extinct. And Neopelina galatheae was not alone. In the decades following that first stunning discovery, more deep-sea expeditions found more monoplacophorans. Different species, yes, but all following the same basic body plan that had been successful 500 million years ago. Scientists have now described more than 30 species of living monoplacophorans, all found in deep ocean environments, typically at depths greater than 2,000 meters, all quietly existing in a world we barely explore, all blissfully unaware that they were supposed to be extinct. Valeropelina gretcheni, discovered in 2022 during the Aleut Bio expedition, but only formally described in the November 2025 publication, is one of these survivors, and it is special for several reasons beyond just being a living fossil. First, the depth. At 6,465 meters, it is one of the deepest living monoplacophorans ever found, pushing the known depth range for this ancient group and showing that they can survive in the Hadal zone, the deepest, most extreme parts of the ocean. Second, the anatomy. Like all monoplacophorans, Valeropelina gretcheni has a cap-like shell, limpet-shaped but not closely related to limpets, and underneath that shell is a body plan that features serial repetition of organs. It has multiple pairs of gills arranged along each side of its body, multiple pairs of kidneys, multiple hearts, or at least multiple atria connected to a central ventricle. This repetition of organs makes monoplacophorans look almost segmented, superficially similar to annelid worms or arthropods with their repeated body segments, even though mollusks are generally not considered to be segmented animals. For many years after Neopelina was first discovered, scientists thought this serial repetition might indicate that monoplacophorans were some kind of evolutionary link between mollusks and segmented animals, maybe representing a transitional form showing how segmentation first evolved. That theory has since been largely abandoned based on molecular and developmental evidence, but the serial repetition remains a fascinating evolutionary quirk, a developmental pattern that somehow works for this ancient lineage even though it is rare among mollusks. Third, and this is huge from a modern genomics perspective, Valeropelina gretcheni is one of the first species in the class Monoplacophora to have a high-quality genome published directly from the holotype specimen. That means scientists were able to extract DNA from the actual individual that defines this species, sequence its entire genetic code, and make that information available to researchers around the world. This is transformative for understanding the evolutionary relationships of these ancient creatures, for placing them correctly in the tree of life, for understanding which genes have been conserved over hundreds of millions of years and which have changed, for reconstructing the ancestral genome of early mollusks. Genomic data gives us a time machine into the Paleozoic, letting us compare the DNA of modern monoplacophorans with that of other mollusks to figure out what the common ancestor looked like and how different lineages have diverged since then. But beyond all the scientific details, beyond the anatomy and the genomics and the taxonomy, the most important thing about Velaropolina gretcheni is what it represents on a deeper, almost philosophical level. It is a time traveller. It is a window into the ancient world, a living connection to an ocean that existed before complex life on land, before forests, before vertebrates left the sea. Its lineage has survived 5 major mass extinctions. The End-Ordovician extinction that killed 85% of species, the late Devonian extinctions that wiped out most armoured fish, the End-Permian catastrophe, the worst mass extinction in Earth's history that killed over 90% of marine species, the End-Triassic extinction that paved the way for dinosaur dominance, the End-Cretaceous impact that finished off the dinosaurs and countless other species. Velaropolina's ancestors survived all of that, persisted through climate shifts and ocean chemistry changes, and the rise and fall of entire ecosystems, found refuges in the deep sea where they could ride out catastrophes that destroyed shallower habitats. And here it is, still existing at 6,465 metres in the Aleutian Trench, crawling across the sediment on a broad muscular foot, scraping up whatever organic material it can find with its radula, filtering oxygen from the near-freezing water with its multiple pairs of gills, completely unaware that scientists spent most of the 20th century thinking it was extinct, completely oblivious to the fact that its existence challenges our assumptions about survival, extinction, and the resilience of life over geological timescales. The 14 species described in November 2025 were not just the carnivorous clam, the popcorn parasite, and the living fossil. There were 11 others, each one strange and fascinating in its own way. There was Spintha bonorum, a tiny polycate worm that lives in the deep ocean and exhibits bioluminescence. Imagine a worm that glows in the dark, sitting on the ocean floor like a living Christmas light, attracting prey or mates or both. The researchers described it as having a cosmic appearance, resembling a living underwater crystal. Then there was Apotectonia senckenbergi, an amphipod crustacean discovered in a mussel bed at the Galapagos Rift hydrothermal vent fields at a depth of 2,602 metres. This one was named after Johanna Rebecca Senckenberg, who lived from 1716 to 1743 and was a naturalist and benefactor whose support for science and medicine helped lay the foundation for the Senckenberg Society for Nature Research. It is rare to see deep-sea species named after historical figures, especially women from the 18th century, and it is a nice touch that honours the legacy of someone who supported science long before women were widely recognised in the field. There was Levedentalium visai, a tusk shell that was actually already known to science, but had never been fully studied. When researchers examined it closely during this project, they found something unexpected. It was carrying a sea anemone attached to the anterior side of its shell. This is the first time an interaction like this has been documented in the genus Levedentalium. The tusk shell had its own hitchhiker, a symbiotic relationship happening at 5,000 metres deep, completely unknown until now. There were also new species of polychaete worms, chitons, and other mollusks, each one collected from different parts of the world. Some came from the South China Sea, some from the Solomon Islands, some from the Gulf of Mexico. The range of depths was staggering. The shallowest specimen was found at just 1 metre deep in the intertidal zone. The deepest, as we have already discussed, came from 6,465 metres in the Aleutian Trench. That is a depth range of over 6,400 metres, spanning almost the entire vertical profile of the ocean. And every single one of these species was new to science. Every single one had been sitting there, living its life, completely unknown to humanity until 2025. This is the reality of marine biodiversity. We have been sailing across the oceans for thousands of years, and we have barely scratched the surface of what lives down there. So why is the Aleutian Trench such a hotspot for bizarre life? What is it about these subduction zones that makes them so biologically rich? The answer is complicated, but it starts with chemistry. When the Pacific Plate subducts beneath the North American Plate, it does not just sink quietly into the mantle. It drags massive amounts of sediment, water, and organic material down with it. As this material gets deeper, the temperature and pressure increase, causing chemical reactions that would not happen anywhere else on Earth. Water trapped in the sediments gets heated and starts to percolate back upward through cracks and fissures in the overlying plate. This hydrothermal fluid is loaded with dissolved minerals, hydrogen sulfide, methane, and other compounds that most organisms would consider toxic, but not all organisms. In the deep ocean, where sunlight never reaches, photosynthesis is impossible. Plants cannot grow, algae cannot grow. The base of the food chain that we rely on at the surface simply does not exist. So life has evolved a different strategy: chemosynthesis. Bacteria that can take chemicals like hydrogen sulfide and methane and use them as an energy source to produce organic compounds, just like plants use sunlight. These chemosynthetic bacteria form the foundation of an entirely separate food web. They are eaten by small invertebrates, which are eaten by larger invertebrates, which are eaten by fish, and so on up the chain. But it is a food web built not on light but on chemistry, and subduction zones like the Aleutian Trench are incredibly rich in the chemicals that these bacteria need. The result is a biodiversity hotspot in one of the most hostile environments on Earth. The Aleutian Trench is not a barren wasteland. It is a thriving ecosystem powered by geology. The same tectonic forces that cause devastating earthquakes and tsunamis are also driving life in the deep ocean. It is a reminder that life on Earth is not passive. It is not just sitting around waiting for conditions to be perfect. It is aggressive. It is opportunistic. It will exploit any source of energy it can find, no matter how strange or inhospitable the environment. And in doing so, it produces creatures that look like aliens. Now let us talk about how all of this happened, because the November 2025 announcement was never just about 14 new species. As exciting as those discoveries are, it was fundamentally about a paradigm shift in how we approach marine taxonomy, about breaking down a bottleneck that has plagued biodiversity science for generations. For decades, marine biology has suffered from what insiders call the taxonomic impediment, a frustrating, persistent problem where the rate of species discovery far outpaces the rate of species description. Researchers go on expeditions, collect specimens from remote locations at great expense and effort, bring them back to museums and research institutions, and then those specimens sit on shelves, sometimes for decades, waiting for someone to find the time and resources to formally describe them. Why does this happen? Why can it take 20 to 40 years, sometimes even longer, between when a new species is first discovered and when it is officially described and published in the scientific literature? The answer is complicated, involving a combination of practical, institutional, and cultural factors that have accumulated over the history of taxonomy. First, there is the sheer amount of work involved in the traditional process. Describing a new species correctly requires a comprehensive anatomical study, often including detailed dissections, microscopy of tissues, measurements of dozens or hundreds of features, comparisons with all related known species to confirm that this really is something new and not just an unusual variant of something already described. That is already a lot of labour. But modern expectations have raised the bar even higher. Journals now typically expect genetic data, DNA barcoding at minimum, and often full phylogenetic analysis to figure out how the new species fits into the tree of life. They expect ecological context, information about where the species lives, what it eats, how it reproduces. They expect high-quality images, often requiring expensive equipment and specialized photography skills. They expect supplementary data files, detailed locality information, sometimes even ecological modeling. Putting together a comprehensive species description under these expectations can take months or even years of focused work. Second, there is a shortage of taxonomic expertise. The number of people with the specialized knowledge needed to identify and describe species in most groups is declining. Universities and funding agencies have increasingly shifted resources away from basic taxonomy, which they often view as old-fashioned or less exciting than molecular biology or ecology, toward more fashionable fields. Many taxonomic experts are nearing retirement with no one trained to replace them, creating a knowledge crisis where entire groups of organisms have only a handful of specialists in the world who can reliably identify them. When one of those experts retires or passes away, decades of knowledge can be lost. Third, there is the museum backlog problem. Natural history museums around the world house millions of specimens that have never been formally studied. Every expedition brings back more material than can be processed immediately. Specimens get catalogued, preserved, and placed in storage with the intention that someone will eventually describe them, but eventually sometimes never comes. The cabinets fill up with undescribed species, scientific treasures sitting in jars, waiting for attention that might not arrive for decades. Meanwhile, some of those species might be going extinct in the wild due to climate change, habitat destruction, pollution, or overfishing, and we do not even have names for them yet. Conservation law generally requires a species to be formally described before it can be protected. You cannot list an unnamed species on endangered species lists or establish protected areas for it. Bureaucracies need official scientific names, need published descriptions, need evidence. An undescribed specimen in a museum drawer has no legal existence, even if it represents a unique evolutionary lineage found nowhere else on Earth. This is the critical gap that the Ocean Species Discoveries Initiative was designed to address, and it represents a genuinely revolutionary approach to solving the taxonomic impediment. The initiative is coordinated by the Senckenberg Ocean Species Alliance, which is part of the Senckenberg Research Institute and Natural History Museum in Frankfurt, Germany, one of the oldest and most respected natural history institutions in Europe. Their goal is simple to state but transformative in implications: make taxonomy faster, more efficient, more accessible, and more visible. How do they do this? By offering a new publication format that focuses exclusively on taxonomy, cutting away all the supplementary expectations that have accumulated over the years. You do not need to include extensive ecological studies. You do not need a complete phylogenetic tree showing how your species relates to every other species in the genus. You do not need experimental data on reproduction or behavior. You just need a robust, data-rich description of the species itself. What does it look like? What are its diagnostic features, the specific characteristics that distinguish it from all other known species? What are its morphological details? Where was it collected? That is it. Everything else is optional, can be published later, can be contributed by other researchers once the species has a name and a formal description. The Ocean Species Discoveries format allows individual scientists to describe new species quickly and efficiently, breaking the decades-long backlog and getting species into the scientific record while they still exist in the wild. And the approach is working spectacularly. The first major collection, published under this initiative, came out in 2024 with 11 new species from various ocean habitats around the world. This second collection, published in November 2025 in the journal Biodiversity Data Journal, includes 14 new species and 2 entirely new genera, representing worms, mollusks, and crustaceans collected from depths ranging from 1 meter in the intertidal zone to 6,465 meters in the Hadal depths. That is 25 new species described in just 2 years through this streamlined process. To put that in perspective, some traditional taxonomic projects focusing on a single genus might describe that many species over a decade or more of work. This is not a bottleneck anymore. This is progress at a scale that actually matches the urgency of the biodiversity crisis we face. The success of the Ocean Species Discoveries Initiative depends heavily on technology, specifically the technology available at the Discovery Laboratory at the Senckenberg Research Institute. This lab is a marvel of modern science. It offers access to integrative research methods including light and electron microscopy, confocal imaging, molecular barcoding, and of course, micro-CT scanning. These tools allow researchers to collect incredibly detailed data on specimens without destroying them. Light microscopy lets you see surface features and fine structures. Electron microscopy goes even deeper, revealing ultrastructure at the cellular level. Confocal imaging uses lasers to create 3-dimensional reconstructions of tissues. Molecular barcoding sequences short, standardized regions of DNA to identify and differentiate species. And micro-CT scanning, as we have already discussed, gives you a complete 3-dimensional view of the organism inside and out. With these tools, a researcher can describe a new species to an extraordinary level of detail without ever making a single dissection. The specimen remains intact, preserved, ready to be re-examined by future scientists using even better technology. This is the new standard, and it is not just about preserving specimens, it is also about speed. In the old days, describing a new species could take months or even years of painstaking work. You had to carefully dissect the organism, sketch or photograph every structure, measure everything, compare it to other species, and write it all up in exhaustive detail. With modern imaging technology, much of that process is automated. The micro-CT scanner produces thousands of images in a matter of hours. Software can automatically measure structures, count features, and generate three-dimensional models. What used to take months can now be done in days. This is how the Senckenberg team was able to describe 14 species in a single publication. They have industrialized the process without sacrificing quality, and they are sharing their methods openly so that other research institutions around the world can adopt the same approach. This is collaborative science at its best, but even with faster methods and better technology, there is still a massive problem. We have explored less than 5% of the ocean floor. Less than 5%. That means 95% of the ocean floor has never been seen by human eyes or cameras. We have better maps of the surface of Mars than we do of the deep ocean on our own planet. And when it comes to ocean trenches, the numbers are even worse. There are only a handful of submersibles in the world capable of reaching depths greater than 6,000 meters, and they are expensive to operate and limited in where they can go. Most of our knowledge of deep-sea life comes from trawling, which is essentially dragging a net along the ocean floor and seeing what you catch. It is crude, it is destructive, and it gives you only a tiny, semi-random sample of what is down there. Think about it this way. If you wanted to study the biodiversity of a rainforest by flying over it in a helicopter and occasionally lowering a net to scoop up whatever you caught, you would miss most of the life in that ecosystem. You would get birds and maybe some tree-dwelling mammals, but you would miss the insects, the fungi, the plants, the soil organisms, all the things that make a rainforest function. That is what we are doing in the deep ocean. We are sampling blind. And yet, even with this incredibly limited sampling, we are finding new species constantly. The fact that a single expedition to the Aleutian Trench produced multiple new species tells you just how little we know. Estimates suggest there are around 2 million marine species on Earth. As of now, fewer than 250,000 have been formally described. That is less than 15%. And many of those undescribed species are probably going extinct before we ever find them. Climate change is altering ocean chemistry and temperature. Overfishing is collapsing food webs. Pollution is spreading into even the deepest parts of the ocean. We are losing biodiversity faster than we can document it. Every species that goes extinct before we describe it is a loss not just for science, but for humanity. We lose potential sources of medicine, biotechnology, and ecological insight. We lose pieces of the puzzle that could help us understand how life works, how evolution happens, how ecosystems function. We lose the stories, and we lose them forever. So why should we care about a carnivorous clam, or a popcorn parasite, or a living fossil mollusk? What difference does it make whether we know about these species or not? The answer is bigger than you might think. Deep-sea organisms are not just curiosities. They are sources of innovation. Take extremophiles, organisms that live in extreme conditions. Scientists have been studying extremophiles for decades because they produce enzymes and proteins that can function under conditions that would destroy normal biological molecules. Heat-stable enzymes from deep-sea hydrothermal vent bacteria are now used in PCR, the polymerase chain reaction that underpins almost all modern genetic research, including COVID-19 testing. Cold-adapted enzymes from deep-sea organisms are being developed for industrial processes that need to run at low temperatures. Pressure-adapted proteins are being studied for insights into how cells maintain their structure and function under stress. The carnivorous clam, Myonera aleutiana, survives at pressures over 700 atmospheres. How does its cellular machinery work under those conditions? What proteins does it use? What metabolic pathways has it evolved? If we can figure that out, we might be able to engineer similar resilience into crops, into medical treatments, into materials science. The same logic applies to every one of these 14 new species. Each one is a solution to a problem. Each one has evolved strategies for survival that we do not fully understand, and understanding those strategies could lead to breakthroughs we cannot even predict yet. Beyond biotechnology, there is also the ecological angle. Deep-sea ecosystems play critical roles in global nutrient cycling, carbon sequestration, and climate regulation. The deep ocean is one of the largest carbon sinks on the planet. Organic material that sinks from the surface gets buried in deep-sea sediments, locking carbon away for millions of years. If we disrupt those ecosystems through deep-sea mining, bottom trawling, or pollution, we could destabilize that carbon sink and accelerate climate change. But we cannot protect ecosystems we do not understand. We cannot make informed decisions about deep-sea policy if we do not know what lives down there or how it all fits together. Every new species we describe is a piece of the puzzle. Every anatomical study, every genetic sequence, every ecological observation adds to our understanding of how the deep ocean works. And that understanding is essential if we're going to manage human impacts on these environments responsibly. Let us circle back to that original provocative question that started this entire journey. Are these aliens? In the strictest, most literal sense, the answer is obviously no. They are terrestrial organisms born on Earth, evolved on Earth from the same common ancestors as every other living thing on this planet. They share the same basic biochemistry, the same DNA-based genetic code, the same fundamental cellular structures. They are part of the tree of life, branches that diverged hundreds of millions of years ago, but still connected to everything else through deep evolutionary time. But in a broader, more meaningful sense, in a way that captures the essential strangeness of these creatures, the answer is absolutely yes. These organisms are so profoundly different from anything we encounter in our daily lives, so thoroughly adapted to an environment we cannot survive in, so bizarre in their morphology and behavior and life histories that they might as well be from another planet entirely. Think about what we have discovered. The carnivorous clam Meionera aleutiana hunts in total absolute darkness at depths where the pressure would kill a human being in less than a second, where the weight of the ocean would crush your lungs and shatter your bones before you even had time to scream. It has evolved a predatory feeding strategy that no other known clam uses, a hood mechanism that creates suction currents to pull prey into its mouth. It lives in an environment where food is so scarce that most animals would starve, and it thrives by being aggressive, by being active, by breaking every rule we thought governed clam behavior. The popcorn parasite Zeioneverta, looking for all the world like someone glued popped corn kernels onto a crustacean's back, lives inside another animal, inside the gill chamber where its host breathes, stealing resources, occupying space, following a parasitic lifestyle so specific and so bizarre that you would reject it as implausible if you found it in a science fiction novel. The living fossil Veleropilina gretcheni has survived for hundreds of millions of years, essentially unchanged, outlasting the dinosaurs by hundreds of millions of years, outlasting the rise of mammals, outlasting ice ages and asteroid impacts and supervolcano eruptions and everything else that Earth has thrown at life in the past 375 million years. It crawls across the sediment at 6,465 meters depth, carrying within its genome the genetic legacy of the early Paleozoic, a time so ancient that the continents were arranged in configurations we would not recognize, and life on land barely existed. These are not creatures we can relate to easily. They do not fit into our mental categories of what animals should look like or how they should behave. They are not cute like mammals or majestic like birds or even vaguely familiar like fish. They are fundamentally other, as alien to our experience as anything from another world. They break the rules we thought were universal. They challenge our assumptions about the limits of life and the possibilities of evolution. And that is exactly what makes them so extraordinarily valuable to science, to human knowledge, to our understanding of what life can be. Science, at its core, is not just about cataloguing what we already know or confirming what we already suspect. Science is about discovering what we do not know, about finding exceptions to the rules and then figuring out why those exceptions exist. Every time we find a creature that does not fit our expectations, every time we discover something that should not work according to our models but does anyway, we learn something fundamental about the flexibility of evolution, the creativity of natural selection, the sheer stubborn persistence of life, finding ways to exploit every available niche, no matter how extreme. The November 2025 discoveries from the Senckenberg Ocean Species Alliance are far more than just a list of new species added to the scientific catalogue, more than just 14 names in a database. They represent a manifesto for how we should approach the study of biodiversity in the 21st century. They are proof that we can study the deep ocean without destroying it, that modern technology gives us the tools to document life in unprecedented detail while preserving specimens intact for future generations. They are proof that collaboration across institutions and countries and disciplines can overcome the bottlenecks that have slowed taxonomy for decades, that streamlined publication formats can accelerate the pace of discovery to match the urgency of the biodiversity crisis. They are proof that Earth still has profound mysteries waiting to be uncovered, still has frontiers that remain largely unexplored, still has aliens hiding in the depths of our own planet that are stranger than most things we could imagine living on Mars or Europa or Enceladus. And most importantly, they are a call to action that should resonate with anyone who cares about the future of life on Earth. We have explored less than 5% of the ocean floor. Think about that statistic for a moment. Less than 5%. That means 95% of the ocean floor has never been seen by human eyes, never been photographed by cameras, never been sampled by nets or corals or submersibles. We have better maps of the surface of Mars, a planet 35 million miles away at its closest approach, than we do of the deep ocean on our own world. We have sent rovers to drive across another planet's surface, sent probes to fly past Pluto, sent spacecraft to orbit Saturn and study its moons, but we have barely touched the deepest parts of our own ocean. And when it comes to ocean trenches specifically, those deep gashes in the seafloor where tectonic plates collide and life exists under the most extreme conditions imaginable, the numbers are even more stark. There are only a handful of deep submersibles in the world capable of reaching depths greater than 6,000 meters. They are expensive to build, expensive to operate, limited in their endurance and range. Most of our knowledge of deep-sea life still comes from trawling, dragging nets across the bottom and seeing what gets caught, a sampling method that is crude, destructive, and gives us only tiny, semi-random glimpses of what actually lives down there. This means there are millions of species we have never seen, ecosystems we have never studied, biological processes we do not understand. Every time we send an expedition to a deep trench, we find new species. Every time. The fact that a single expedition to the Aleutian Trench produced multiple new species, including a depth record for a carnivorous clam and a living fossil from a class thought extinct, tells you just how little we actually know about what lives in the 95% of the ocean we have never explored. Current scientific estimates suggest there are around 2 million marine species on Earth. As of now, fewer than 250,000 have been formally described and given scientific names. That is less than 15%, and here is the terrifying part. Many of those undescribed species, possibly hundreds of thousands of them, are probably going extinct before we ever find them. Climate change is altering ocean chemistry through acidification, changing water temperatures, disrupting current patterns that have been stable for thousands of years. Overfishing is collapsing food webs, removing key species and causing cascade effects through entire ecosystems. Pollution, including plastics and chemicals and agricultural runoff, is spreading into even the deepest parts of the ocean, places we thought were too remote to be affected by human activity. Proposed deep-sea mining operations threaten to physically destroy habitats that took millions of years to develop. We are losing biodiversity faster than we can document it, faster than we can understand it, faster than we can appreciate what we are losing. Every species that goes extinct before we describe it is a permanent loss, not just for science but for all of humanity. We lose potential sources of new medicines because many deep-sea organisms produce unique biochemicals to survive in extreme conditions, and those chemicals might have pharmaceutical applications we cannot predict. We lose potential insights for biotechnology because understanding how life functions under extreme pressure and temperature could lead to advances in materials science, enzyme engineering, industrial processes. We lose pieces of the evolutionary puzzle that could help us understand how life adapts to changing conditions, information that might be crucial as we face our own changing climate. We lose the stories these species could tell us about survival, about resilience, about the incredible flexibility of life over geological timescales. And we lose them forever, because extinction is permanent. There are no do-overs in evolution. Once a species is gone, that unique combination of genes, that specific solution to the problems of existence, that particular branch of the tree of life, is lost for all time. Future generations will never know what we lost because we failed to study it while it still existed. So yes, we found aliens at 6,000 meters deep. We found carnivorous clams and popcorn parasites and living fossils. We found creatures that should not exist according to our assumptions but thrive anyway. We found evidence that our planet is far stranger and more wonderful than we give it credit for. We found proof that modern science can study these creatures without destroying them, can accelerate the pace of discovery to match the pace of loss. And we found a reminder, urgent and undeniable that the deep ocean is not a dead zone or a barren wasteland, but one of the most biologically diverse and scientifically important environments on Earth. The Aleutian Trench, the Mariana Trench, the Peru-Chile Trench, the Java Trench, all the deep ocean environments we have barely touched, these are Earth's last frontiers. These are the places where mysteries still outnumber answers, where discovery is not just possible, but inevitable if we make the effort to look. And the aliens, stranger than most science fiction, more bizarre than most imagination, are waiting in the darkness, living lives we can barely comprehend, holding secrets we have not yet learned to ask about. The question is not whether more aliens exist in the deep. The question is whether we will find them before they disappear.